to introduce Dr. Greg Pask from biology at Middlebury College to talk to us about his research. And I did see lots of pictures of students in there, so you'll get to see a little bit of the inside scoop of student research here. So join me in welcoming Dr.
Thank you so much. Can everybody hear me all right? All right, cool. Well, if you can't hear in the back, you are welcome to come closer. We all know how this works in a classroom setting. So yeah, I'm Greg Pask. I'm here from Middlebury College. A lot of research on insects and specifically how they can smell, hence the social sense of ants and fireflies. So I was asked to talk a little bit about my journey and kind of when I was in your shoes to start. So I went to undergrad at Middlebury College. It's in eastern Pennsylvania, just in Allentown, Pennsylvania, just an hour north of Philadelphia. And I was a biochemistry major. I had nothing to do with insects. I had nothing to do with anything neurobiology, sensory biology related. And I was doing chemistry research there. And then I went to Vanderbilt. I took one course when I was in undergrad that was very interesting. It was neurotransmitter receptors, structure, function, and that kind of started me off on that track. And I wanted to pursue something in that, so I went and I did a PhD working with malaria mosquitoes and their sense of smell and ability to find a host and get a blood meal. It was at that point that I kind of really, we were working with mosquitoes, but that wasn't really I wasn't really that big into insects. And they had a poster that I saw like this. This is the one for this year. But it was an insect chemical ecology PhD course, which was super interesting. And I was like, oh, that should be fun. It was over in southern Sweden in Alnarp. And I went there, saw kind of insect pest management in practice. Went out to apple orchards where they were using all this research to help save their crops. Got to follow a bunch of German shepherds who were trained to sniff out the pheromone of a bark beetle that was really causing problems, and then we followed this dog with GPS collar. So it was super cool, really opened my eyes to the potential for insect smell research. So with that, I pursued a postdoctoral fellowship at University of California, Riverside in the entomology department. That's where I first started really getting into insects. And I started picking up ants, and you'll see those a little bit later. And then I was like, I knew a couple things. I wanted to get into a position where I got to do a lot of teaching. I also wanted to be back on the East Coast. That's where I'm originally from, New Jersey. And I kind of started to do that. I went to Washington and Lee University and was a visiting assistant professor there for a couple years. Then was at Bucknell University where I started up a lab, but then wanted to kind of change scenery after 3 years and found myself in the great state of Vermont here at Middlebury College. So that's my journey, and feel free to ask me any further questions about this. But I've always I think it's really important to highlight that I was doing chemistry research as a biochemistry major, and now my mentor in that process can't believe that I'm doing insect molecular neurobiology at this point. So you're not restricted and confined to what you major in your undergrad days. So I like to work on smell, and maybe you think about your own sense of smell sometimes. You probably don't think about it too much. It's often considered the fifth sense in that it's like the last one that we think about. When we think about the words, when somebody loses a certain sense, oftentimes losing your sense of smell, your olfactory system, is not one of the words that a lot of people know, which is anosmia. So there's it's a big kind of major kind of quality of life issue when you can't smell, because all the things that we associate sometimes with taste are really our olfactory system and all that flavor. But it's something that we've actually been thinking about a lot more, especially as COVID has had impacts on people's ability to smell. And they're like, oh, I can't really taste this anymore, but they're really just talking about their sense of smell. So it's big on people's minds, but it's not as big as it is on insect minds, where smell is a much bigger and more important sense for them. And they don't hide it. So this little Saturnid moth here, you can see those antennae are big, and it's just like Little Red Riding Hood. It's all the better to smell you with. All that surface area where they can interact with different chemicals in the environment Maybe it's pheromone, maybe it's a place to lay their eggs, all sorts of fun things like that. Go back to my mosquito days. We can think about the sense of the odors that we give off and the mosquito's ability to find that and locate us in a crowd. And maybe, like, everybody has their mosquito stories of mosquitoes seem to like me better than X family member or friend. But everybody's getting bit, especially right now in Vermont. One little example of like how we can take advantage of this in a pretty sustainable like insect pest management way is if you've ever gone to an apple orchard, which we're getting into apple season now, you might see these little red plastic ties around trees, and these are actually pheromone dispensers. They are emitting and releasing all of these pheromones for a specific pest species. Most of the time it's for the apple codling moth. So normally you'd have a male who would be trying to train in and follow and locate a female who is calling. She's releasing a sex pheromone out there to attract a mate, and the male zeroes in on her, finds her, and then they mate, and they have, you know, beautiful moth babies in your apples. When you have these little pheromone dispensers in there, there's so much sensory overload that they might either think they find the source of a female and they find this big hunk of plastic. They can't mate with her. Or they just get so overwhelmed and confused that ultimately they just can't locate anything because the scent's just around them and you can't kind of find where the source is. So this is a very good practice as opposed to something like a broad spectrum insecticide, which might just target all of your beneficial, you know, pollinators and predators that help you manage these pests. So it's very non-specific, where something like a pheromone is only targeting something like the codling moth. So, you know, we have these nice, beautiful antennae here. This is an eastern black carpenter ant. And you might think that, all right, they just have kind of little sticks that come out of their heads as their antennae. But when you look closer, they look much cooler. This is an image from one of my students here, Hannah. This is the tip of the antenna, and you can see, even though it looks kind of smooth up there and, you know, like a stick, it's covered in all these different hairs. And that is how this ant is able to interact with the chemical environment. We zoom in a little closer, and these are all scanning electron microscopy images here. We see one of those hairs looks like this. And I know it's a little bit hard with the lighting to see, but can you see those little dimples at the tip? Those are the pores. These are like the nostrils of the antenna where all the odors come in, and they're able to detect them. So we can turn that into a cartoon here, which I've done here. And you can see this odor plume comes in, and it moves through these pores. And then we have all of these olfactory receptor neurons, or ORNs, that kind of put up their receiving side of the cell into these hairs. And that's where all these molecules get detected and get picked up. So on the surface of those neurons, you have these, my favorite set of proteins, the odorant receptors. These are the specific things that say what an insect can detect or not, what molecules they are sensitive to in their environment. So if we zoom in on that, we see this odor coming in, this nice little plume, and it moves in. And we see something that is familiar for a lot of kind of like membrane and cell biology. We see an ion channel here. So this odor comes in, it binds to what is known as an odorant-gated ion channel. It binds to it and activates it, and all of a sudden that channel opens up, ions move in, and it activates the cell, and all of a sudden the brain is like, I smell peaches, or whatever that smell could be. So this is where I like to live. These ion channels are made up of two different types. One is known as orco. This is like a highly conserved part of the protein, part of this channel that is, you can take an orco from an ant and swap it out for one from a fly and it still works, and vice versa. And it's required. If you don't have orco, you get an anosmic insect and it can't smell its environment. So that's the kind of more consistent, stable part of this grouping of ion channel subunits here. And then you have what we call the ORX, and this is where you get the highly variable, the most interesting part of what you'd imagine for understanding the olfactory system. We have a lot of different proteins and genes that lead to this, And these are where you get the things that are specific to a certain odorant. So if you have a receptor for the smell of peaches, you have a receptor for the smell of the sex pheromone of your species, a receptor that specifies like a harmful microbe that's on a place where you might find food that you want to avoid. So this is where you get all that diverse sensory function and increased high definition sensory system action. Okay, so the first story I want to tell is kind of centered on the ORX side, where this particular ant species really has a lot of them, and almost as many as we do. So ants are very successful. You might find them in your house. They're being pretty successful there. You might find them out in the wild, and they are thriving, right? It's a model. It's a model species for success. Model grouping of all these ants just have great success because they work together, right? It's this motivational thing. You can make all these nice posters of it. These are weaver ants. They are physically working together and holding these leaves together to start to weave their nest up in the canopy of trees. They even get the young involved in this process. This is a little larva here, and the adult weaver ant is using it essentially as a glue gun. As these leaves are being held together, the larva can spit out some silk and kind of adhere these leaves together. And it's all really successful. So you got adults, larvae, all working together for this common cause. And then you get like motivational posters that people put out like this, where it's definitely fabricated, right? Ants don't use their front legs to hold up sticks, right? That's what their mouth parts would be for. But it gets that message across that ants are really a model for teamwork. And all of this teamwork really relies on what we're interested in, chemical communication. Being able to talk with each other, right? These ants aren't communicating through their spoken words to work together here. They're using chemicals that signal very reliable messages to their members of their species or their colony. So they might have a nice little trail that they're all following. Maybe it's to find a new nest. Maybe it's you left a bunch of crumbs out on your kitchen counter. And that's what they're locating and telling everybody, hey, there's more food here. And they continue to lay a trail pheromone down along the path, and then you get big lines of ants crawling. And that's how you get ants. Or you might see these ants here. They are looking antsy, looking to defend their colony. They have the stopping power to kind of stop a bear in its tracks. Right from their end of their abdomen, they're able to curl it up and spray acid at great distances into the eyes of a bear that might be kind of digging up their colony and whatnot. But they're also communicating with each other because that acid, that formic acid that they spray, is an alarm pheromone. And all the other ants smell that and say, oh no, there's something wrong, let's defend the colony. And then you get more ants spraying more acid in a more angry bear. You can also have these eggs in a colony, and you want to know, are these eggs laid by my queen, or are they laid by some invading parasitic species that's trying to take advantage of our workforce? And you're able to do that because those queens, or those eggs might smell specifically of that queen's scent. And then last but not least, something that maybe you've seen on the sidewalk walking around. You see two ants interact. Are they from the same colony and they're working together and maybe they're going to share some food? Or they interact and they're not from the same colony. Maybe they're trying to compete over the same resources, and then fisticuffs ensues and they're grappling, trying to sting each other, all good things like that. But it's all this chemical communication. And this is where I want to introduce our favorite ant, the one that we have in my lab at Middlebury. This is the Indian jumping ant, also known as Harpagnathos saltator. The translation of this is the sickle-jawed leaper, named for these large mandibles, and then they actually do lunge and jump to strike, to grab prey and sting it. We really like this species. They're nice and big, around like 1.5 centimeters long. Easy to take care of. We sequenced the genome, which gives us a lot of access to all the different inner workings of their olfactory receptor genes. In the lab, they're actually easier to take care of than fruit flies, which are like a common model system. We just kind of bring them these live crickets. They sting them, which paralyzes them. They bring them down, and the larvae and the adults suck out all the juices from the paralyzed cricket. And then to take care of them, we just kind of scoop out the carcasses of the dead crickets that they pile up in the corner, and that's about it. So very easy maintenance. But the thing we really like about them is this very unique social structure in which they are different from most of the ant species we interact with. It might be a nice stepping stone to learn something about how these ants are working together and communicating. So for the reproductive biology of this, here's our queen, denoted by this nice crown that I put on her up there. And then we have a worker here, and typically a worker will live around a year as for its lifespan. They would like to be reproductive, right? You know, laying eggs is cool, passing on your genes is good, but the queen's there, so that signal that she is giving prevents that worker from laying their own eggs. If she does, there's also other workers that'll start to police her. If they find eggs that are not laid by the queen, they will eat the eggs. They also might detect the reproductive nature of this cheating worker, grab her, carry her around, and be very aggressive to her until she stops this, insulting behavior to the colony. And this is what this looks like here. You can guess which ant is the one who is trying to lay some eggs and the other one that's just not having it. There's a lot of grappling, and sometimes you'll see kind of one of these ants grabbing one and carrying one around, and you think, no, maybe it's just carrying a dead ant around, but it's actually policing it in progress. And then you open the lid, and both of the ants go their separate ways and walk away from each other. So, yeah. You can see how big they are. This is relative to kind of a smaller cricket hanging out in the corner and a little larva that's eating it up. But if you don't have the queen around anymore, she dies, she's waning in her reproductive status, or artificially in the lab we remove her, then all those pressures go away and we hope to get some sort of fully reproductive individual, which is termed, unfortunately, it's Gamergate, which was out before Gamergate occurred in like the video game industry. But you have this big transition that a worker with the same genome is now fully reproductive, lives 3 years as opposed to the 1 year originally, and is able to kind of carry on the reproductive duties of the colony. So most ant colonies, the queen dies, they might send out males and unmated queens elsewhere, but these colonies, when the queen dies, they stay, and so they're kind of immortal in that way, that they continue to survive, they make their own reproductives, and they're able to manage that indefinitely as new workers come in to the picture. And they decide who it becomes the reproductive because of a lot of dueling interactions, this kind of battle royale, but nobody dies, which is nice. So these ants are also really big, so we can paint them with little color-coded dots to know what individual is who. And this is what happens when we have workers engaging in a lot of antennal dueling. They look like siblings fighting, which is exactly what they are. And you can see here's some other ants that are kind of just taking care of the brood, while these ones are fighting, kicking around some of the eggs that are on the ground. So at the end of this, somebody becomes the reproductive, and we move forward. So it comes to this really interesting question of, like, say this is you're this ant in the colony, and kind of one of the mindsets you have as an ant in the colony is you have to respect the Gamergate. Who is that reproductive queen? Because she looks exactly like you, because she used to be you as a worker. So there's no visual signals there, even though these ants are highly attuned to their visual system. You can sense by the big eyes relative to their head. But we have this kind of mindset here. This is a graphic that my wife, who's a really talented digital artist, made for me. You can imagine it's like a poster that's hanging up in the colony that you have to respect the Gamergate all the time. So it's a question of which one is the Gamergate? How can you tell who is the reproductive in your colony? Any thoughts? What was that? The pheromones, yes, the sense of smell, yes. One of these smells like the queen, and one of those does not, or the pseudo-queen, if you will. And it all comes down to these little molecules like this. If anybody has taken organic chemistry or will be taking organic chemistry, these are cuticular hydrocarbons. These are hydrocarbons. There's only carbon and hydrogen here. Sometimes there's some double bonds in there. Sometimes there's some methyl groups. And the longer ones signal who is the gamergate and who is the worker. So you can also imagine in that transition from worker to gamergate, all of a sudden these that are all around the outside of you get a little bit longer. And that's how it's signaled and reflected. It pairs well with your ovarian activity as an ant. So these CHCs, these cuticular hydrocarbons that are coating the outside, that all these social insects have used as recognition pheromones to figure out colony, species identity, colony identity, and then what your role is within that colony. Are you reproductive? Are you a forager? Are you a nurse ant? All these different roles can be decoded in this. I like to think of them as little barcodes that other ants can read off of you and figure out what you do and what's your role in the colony. What we were really looking at is all these different cuticular hydrocarbons of all these different chain lengths and unsaturations and branching patterns that come off of them. So this is a gas chromatograph showing all these different peaks are a different type of molecule. And how tall that peak is kind of how abundant it is. And you have different changes that signal the differences between certain colonies and whatnot. So because these are kind of different, once these ants would interact, all of a sudden now they're probably going to fight in some way. Yes?
Can you tell which one is the gamergate?
So
With those charts?
yes, you can. So I'll show that in a little bit, but for like that particular species, these ants actually don't have gamergates, but they have just true queens, these ones here that I'm showing. It's just such a good image. But yes, you could pick up these, you could kind of analyze the molecules that are in these different kinds of traces and be like, oh, there's actually more molecules, higher abundance of ones kind of further out that denote those really longer and larger molecules. That tells you what it is, yeah. All right, so this was known really far back. The ants are really good at this. I'm a huge nerd. And, from Lee and Kirby, 1962, the second appearance of Hank Pym, the first appearance of him as Ant-Man. He's in the colony and he says, oh, they're turning around, they're coming this way, but how did they? Oh no, I forgot. I forgot that they have a highly developed sense of smell. They're able to detect any alien creature near them, right? So this was known in the field, that ants are able to kind of do this very nice detection. You drop another ant in and into the wrong nest and there's problems. So we try to channel a lot of this in a lot of the things we do in our lab. So one example, this is some work that I'd done with students on like a little side project in a class, in a kind of like a 4-week class on insect chemical ecology. We have a little ant here and we have a glass bead, and we coated that glass bead in just the cuticular hydrocarbons of a non-nestmate. So this is not somebody who's from their colony. And this is our artificial ant. And if we watch this video, you see it's enough to trigger a response. So it finds this bead, doesn't smell like a member of its colony. In fact, it smells like an invading member of another colony. And it is trying to bite it. This lasts for like 5 minutes and still just as aggressive. It's trying to bite it. It's trying to curl its abdomen and spray acid on it. And even though these ants have visual abilities, even though it looks like a bead, it smells like an ant from another colony. So the CHCs are very powerful cues for them. In our favorite species here, we talked about these gamergates, these reproductives, whether it's a queen or one of these pseudo-queens, have these longer chain hydrocarbons. One particular hydrocarbon, this is 1323 dimethyl C37. So it's 37 carbons in a row. And then at position 13 and 23, there's a methyl group. This molecule is not found in outside foraging workers, inside nurse workers, unmated queens and dispersing queens. But then once you have a queen that is reproductive, it starts to appear. Same thing with gamergates, which used to be workers that now ascended to the throne. So this is a molecule that we think is a candidate fertility signal. It's signaling to the other ants, my ovaries are active and I am reproducing for the colony. So that's a molecule of interest. But there's all these questions like what genes are actually responsible for detecting these very important cues that hold the colony together. So that's where we started, and that's where we wanted to focus on this ORX, this odorant receptor that we have all these genes, now that we have this genome for this ant species to look for. And our first, so this is a big phylogenetic tree of all the odorant receptor genes in a bunch of hymenopterans, which are bees, wasps, and ants. And we see all of our ants are like the nice fancy colors. The honeybees and the wasps are like the purple and the black here. And this ant species, Harpynathus saltator, the Indian jumping ant that I was showing all the pictures of and the videos of, 347 olfactory receptors, which is a lot. We have about 400 as humans. So it's a lot, and it's the most that has been found in any insect species. Ants just have way more than like a fruit fly. This grouping really stands out for us, this 9-exon subfamily, because it contains about a third of the entire ant receptor or like receptor repertoire. So of the 347, 118 of them, so if we look here at just Harpynathus saltator, this red represents all the 9-exon subfamily. So that was like, oh, there's a lot of kind of evolution happening in that subfamily. That might be a good target to find these pheromone receptors. They also have a lot more enriched expression compared to like workers express them a lot versus males. And I haven't talked about male ants, but male ants don't really do anything other than just mate. They have wings, they're haploid, they come from unfertilized eggs, and they live about a couple weeks. I kind of think of them as like flying packets of sperm, and that's about it. What was that?
Couple weeks.
Couple weeks, yes. We're actually in that time period for some ant species right now, though. You'll see some winged ants around, a lot of winged queens that'll be flying around. They'll mate with these male ants briefly, maybe multiple times. The males die away, and then the queens will actually chew off their wings. And I've actually seen this happen like on the window of my car, of like wings just sloughing off, and then the female will just find a place to tuck in and start founding a colony. So yeah, and they store that sperm. Those queens might live anywhere from like 15 to 30 years. They keep that sperm from that first mating around and alive in a special organ in their body.
This is a different species?
This is all ants.
All ants.
Most ants. There's you always get really cool, like there's some that are asexually producing and they break all the rules. But yes, the sexually reproductive ants, they store that and keep that sperm alive in an organ called the spermatheca. So yeah, just a little fun. So I'm a bug dork, so I want to bring these insights to you. So we have this kind of targeted family that we wanted to look at of these genes. And that's a good place for us to kind of think about using them. So we do a lot of molecular biology that I'm not going to get into because it's complicated and kind of niche. But we'll kind of grind up antennae from ants and then clone out our genes of interest, put them into all sorts of different plasmids, grow them up in bacteria, so use these bacteria as plasmid factories to grow up our genes of interest. And we really want to just get them to a fly. Because when we do this, when we kind of try to do this work in ants, we don't have the genetic tools to really make a lot of simple decisions like, oh, this receptor responds to this molecule, because there's a lot of complexity there. We kind of really want to deconstruct the system, break it up into small parts, and see how each of those parts works. So when we take these 23 ORs that we looked at from the 9-exon subfamily as like our first pass, we went there, we put them into a fly in a genetic construct known as a UAS fly, and I'll get into what that is here. So the Gal4 UAS system is this binary expression system that's a really powerful tool in Drosophila. All of these things, you have kind of a Gal4 fly and a UAS fly. I like to think of them as the where where do you want this gene to be expressed? And that's your GAL4. That's the cell-specific part of that construct. And then the UAS is the what. What are you trying to express? In our case, it's an ant receptor. It's an ant olfactory receptor gene. So we cross these two flies, and we get one fly that has both parts, right? This is a binary expression system. When you don't have, one or the other, it doesn't work at all. Also makes for really nice controls. So the cool thing about this is flies don't normally have GAL4 genes or UAS genes in them. These come from yeast. Way back in the day, people engineered these genes into flies to do the things that we get to do, to kind of have this nice genetic control in the system. So GAL4 is a transcription factor that's going to start the expression of certain genes in the fly, well, in the yeast normally, and that transcription factor will bind to this upstream activation sequence and promote the expression of that gene. So what this looks like when we have a fly that has both of these components, in every cell that would normally express orco, that really important olfactory gene, so we're just targeting the olfactory system of the fly here, we have this orco promoter upstream of a GAL4 gene. So in those cells, we get the translation of this GAL4 protein. That GAL4 protein likes to bind as a transcription factor to this UAS gene, and that's going to now promote the expression of our ant receptor gene inside the fly. So we have our ant ORX here. Remember how I said this orco is kind of interchangeable across different insects and it works the same way? It's kind of really required and conserved. We can just rely on the fly orco to work with the ant ORX to get some sort of functional response. All right, so now we have flies that are expressing ant receptor genes in exactly the neurons where we expect to find them. And now what do? This is one of the fun things I like to do. I like to poke tiny little electrodes into fly antenna. So we take a fly, we immobilize it, which means I use a little mouth aspirator or pooter to suck the fly in and then shoot it into a little pipette tip, and it gets all stuck in there with its head poking out a little bit. We backfill it with clay so it doesn't escape, and we kind of poke it out a little bit so that just kind of half the eye and the head is sticking out. And then we pin down one of the antennae that we want to have access to. We then take one electrode and stick it in the eye as our reference electrode because the eye should not be responding to different chemical cues that we puff on. And then we take the other electrode and we stick it into a certain hair on the antennae of this fruit fly. So you've all seen fruit flies before, probably at your fruit bowl. So think about their size of their antennae, and then think about the size of one of the hairs on their antennae. And that's the system here. And then we have that, and we puff an odor onto it, and we want to see what kind of response does it give. Does this ant receptor that we've now expressed just by itself in a fly, does it give the fly a new sensitivity to a certain odor that it didn't respond to before? Now we're starting to bridge the connection and start decoding how these receptors work. All right, we just look at the neuronal activity while we puff that. I have a little video here. On the bottom, you'll see when we puff the odor. And these are all the neurons firing in real time. We get to see it. After we puncture the hair with the electrode, we start to see these spikes happening. And we have a guitar amplifier hooked up to the system, so we get to hear the spikes as we do it, because you're looking at a microscope, and being able to hear that allows you to know, all right, I've hit pay dirt, and I can start recording. And we'll puff the odor now, and we see this nice increase in spike frequency, which means that that neuron that now expresses our ant receptor, that receptor opened up, all those ions flowed through, and the neuron got activated and starts firing all these action potentials, and we see that response. If it didn't respond, nothing would happen. It would just look like this little baseline signal here. So we do that for one of our receptors here, HSOR281. We puff just a blank with our solvent, our vehicle here, and we get no response. But then we throw in C30, so 30 carbons in a row, this nice linear hydrocarbon, and we get this nice big response. So now HSOR 281 responds to C30. We can do that with a bunch of other ones. We find something really interesting that it's very specific, right? C29, nothing. C31, nothing. So you take away a carbon or add a carbon, and this receptor is not going to respond to it. It's very narrowly tuned to this specific odor here. Another thing, just because we are all aspiring scientists here, that controls are really important, right? So we can do that. We can take out the whole UAS component, right? This binary expression system needs both things to be there to function. We take out the UAS and say, all right, the flies that have this exact same genetic background, but they just don't have this other component here, they don't respond to any of these molecules. So this is not something that a fly can natively respond to. It's because we've added this, so we get this nice gain of function response. We've added this ant gene, we get this new response. Therefore, this response is related to that ant gene. Does that make sense?
You're
Cool.
saying it would not have responded before, but
Yeah.
now it does.
Flies don't care about this molecule, yes. But then we put in a ant receptor gene that responds to it, and they're expressing it, and it's all over their sensory neurons, and now they can respond to it.
You're traced to say you had an effect.
Yes, yeah. And we don't really care about the behavioral, like what does that do, because that just depends more about like where we put that gene and like what the wires are in the brain for that. We're just looking at staying at the periphery, seeing, all right, we kind of use flies as like a cell culture system to kind of like, all right, let's have this blank slate here. Let's put our gene in and see if it increases the response to something. So the one thing that is simple to point out is that this odor panel is kind of boring. These are just linear alkanes. These are just linear hydrocarbons. This is also what you can purchase easily and readily. When I was at UC Riverside, we had all these ants as well. And we wanted to kind of like, all right, what are some like natural extracts that we could take off of these ants? Because it's not just these molecules here. We want to see all the things that are on them. So we took ants that were gamergates, workers, and these are what the males looked like, right? They don't look like much of anything. Haploid, only one job, mating and then dying. And we took extracts, we washed them with a solvent and took all the hydrocarbons off their bodies and puffed them onto our flies that expressed a certain receptor. So we can do that for a couple here, and we start to see some differences. That the worker extract and the gamergate extract and the male extract gave kind of moderate responses here, so probably odors that are shared, hydrocarbons that are shared across all the members of the colony, versus something like HSOR 263, which we have this nice big response, high frequency firing here, to this gamergate extract, and not really much, small responses, but not anything big from the workers. So it was very exciting for us. It was one of the bigger responses that we got in this kind of screening panel, and it was something that we were very interested in, especially with that molecule of interest for us, that fertility signal that we wanted to see. So we wanted to focus in on like, hey, what is specifically in the gamergates that is activating this gene? This is an older paper from 2000 that looked at all the hydrocarbons on this ant species body. So we can see these are all those linear alkanes here. These are just, C28, 28 carbons in a row. C30, the friend that we saw the response from before, is there. But then we see all this additional complexity and diversity from all these other species, or all these other chemical species on their bodies. We get methyl branching at different positions. We get double methyl. We get little unsaturations, double bonds that are breaking into these structures. And the one that I want to point out here is this one. This is that one that we saw before that was only present in the reproductives, the gamergates, the pseudoqueens, and the true queens. And that's one that we were really interested in, but you can't buy it. Thankfully, Jocelyn Miller, who a collaborator that'll come up later that I still work with, was right down the hall at the time. And he had all these different, he's like a top insect synthetic chemist for pheromones in the world. And was like, yeah, I can make that for you. C37, two methyl groups, I can make it. Made this in like, a week or two. Gave it to us. But because he was such a big deal, and other people interacted with him for their pheromone needs and cuticular hydrocarbon needs, he had a bunch of other molecules that he could just he made them for another project. And he was like, yeah, take some of these too. Get your pick of the litter here. So we did that, made this molecule for us. We tested it individually, and we saw this huge explosive response from this gene. So that response that we had to the gamergate extract was indeed related to this molecule that is this candidate fertility signal. So that was very great to see. We also saw that some of these other molecules that he was able to lend us that are also in the ant, or on the ant, gave some responses as well. So it kind of diversified and increased the realism of our little screening panel of cuticular hydrocarbons. So this is an immense amount of work that's shown really small that you can't really see it here. But I wanted to highlight, kind of looking at all those 23 ORs that we were looking at, something like this, HSOR 276, we start to understand some of the logic behind how these receptors work. So something like C28 gave a response, C29, small little response here. But if you take one of the carbons off of C29 and then stick it one carbon position in, you get 2-methyl C28. And that seems to increase that response. So then we can start to think about how this might work in what that binding pocket is on that receptor. Other receptors kind of had more of a broader tuning. Anything from kind of C32 to 35 gave some responses, and then these methyl branch C31s. So maybe these are the right odorants, or maybe there's one that really gives a big response that's just not in our screening panel. So we did that for all these. We start to kind of put them all together. Another representation of the previous data that I wanted to show. We start to see some trends that kind of below C27, like C27 and below, we're not really seeing any responses to these genes. So that the 9-exon subfamily that we focused on is kind of all responding to these larger hydrocarbons. But some of these other ones are found in ants and other ant food and bits like that. Maybe some other receptors from a different group are responsible for responding to those kind of molecules. So to wrap up that kind of this first part of the story, this 9-exon subfamily was like the first ant pheromone receptors ever looked at and kind of identified. It's the first long-chain hydrocarbon receptors of any insect. People have been mostly focusing on much smaller things in the 20s, low 20s. And we kind of think that it's probably binding somewhere between the membrane region of this receptor because it's so hydrophobic. These are waxy molecules. So we kind of think that they're fitting in some way here. And something in the receptor itself is detecting, all right, this is the right amount of carbons in the right position for us to respond to this and get nice, to activate us. It seems to be coded combinatorially, which is something that we have, where we have many, if we go back here, we have some molecules that activate many different receptors, and receptors that respond to many different molecules. You put both of those possibilities together, and you get really good high definition acuity in your olfactory system. So that seems to be something that's true across all these other eusocial insects. And this pairing, this 1323-dimethyl C37 and HSOR 263 is this kind of interaction we really want to explore further to see, hey, maybe we can knock out this gene and all of a sudden ants start to behave and they don't respect the gamergates at all, right? Kind of disrupt the whole colony structure of the species. So some of our future work, we continue to do this kind of, looking at certain expansions and really rapid evolution of these gene families. And can we take a really closely related group of these genes and make small little changes, maybe make chimeric swaps, which means taking one region and swapping it with another and seeing any sort of change of function. Start to understand how this all works. We have different amino acid sequences that we can start to play around with. Like these two have this RSH region, but these other ones don't. Maybe we swap that and see how it changes the molecules that it detects. So we can start to get into the nitty-gritty, the nice like structure, function, biophysics of it all, which is the place where I like to be. But we go back here, and I've always really been excited about kind of the organismal and the ecological side of this, the chemical ecology. And that's where we step into this firefly that we're going to talk about and kind of what potential pheromones might be there. So many of you probably don't think of fireflies as like pheromone focused, right? They're flashing things at night and whatnot. I like to introduce you to the winter firefly. This is a firefly that's very popular and abundant in this area, and it's one that I actually think should be the next state insect, the new state insect of Vermont, and dethrone the current state insect. Does anybody know what that is? Or any guesses? See, a bumblebee would be cool because it would be a native species. Instead, our state insect is the European honeybee, which is quite lame. So, I mean, agriculturally it's important, but that'd be like having the cow as our state mammal, right? So, which, I mean, maybe people are for, who knows? But there's some reasons why I think this is a great representative of Vermont. The first is that the winter firefly toughs it out during the cold months just like we do. Doesn't feel like it now in the heat wave. But most fireflies, they are out as adults, and that's when you see them flashing in the summer. They lay eggs, and then the larvae are underground, underneath the frost layer, you know, kind of resting, and then they'll start eating when it gets warmer later. And then they come out, and they have very short-lived time as adults. The winter firefly kind of flips the whole life cycle on its head, in which they are actually, I saw my first one coming out in the fall, or it's end of summer now, not fall yet, last week. So we're kind of like in this phase, they're starting to emerge. And then they'll tough out the whole winter, kind of in cracks of bark, underneath logs and rocks, and just ride it out. And then you can like warm them up in your hands, and they're fine. Here's an example. I partner with the Bread Loaf You Farm, which is a maple farm in Cornwall, Vermont. And this is an old maple sugaring tap hole that one of these winter fireflies has poked its head in and is just resting in there for a little bit. Other thing is that winter fireflies really like maple sap, right? Nothing says Vermont like that. You can find them, you know, circled around sap flows on maples like cattle around a trough. That's what maple sugarers will say. So I've gone to, maple farms. You can go into the old school buckets, and you see winter fireflies floating on the top. Like every maple sugarer I've talked to knows the species, and they know the species well, because when they're coming out, starting to be active in like the mid to late spring, there's not many sugar sources out there. Maple sap is a great source, and when we are tapping into that source in an abundance, they also can exploit that as well. Maybe you've seen the more modern tubing collection of sap from maple trees. Any little micro leaks in there, and you'll see like, that's what this is talking about, like any leaks of sap out of that tubing, you'll see winter fireflies gathered around all drinking it up. So yeah, they really like sap, they like the cold winter. And they're really a nice model for good conservation practices here. Firefly-friendly lighting practices is something the Xerxes Society for Invertebrate Conservation puts out there. Winter fireflies, just like us, have turned off their lights, right? A lot of people will talk about, there's not as many fireflies as there used to be, and then I'll go to their house, and they have big floodlights coming out of their house when nobody's around. They live out in the country. And that's just light pollution for all these fireflies that are trying to mate in that space with the light. So winter fireflies, you look at them, you're like, that's a firefly, bona fide firefly. Then you flip it over and you're like, where's the light organ? Where's that bright yellow light organ that I'm used to seeing? And they don't have one. They have lost the ability to produce light as adults. Instead, what do you think they use? Pheromones, smell, right? Just like all the other insects do. The fireflies are the ones that are out there in the extremes. So this is our winter firefly, no light organ. They are still fireflies. They do glow as larvae as a signal of don't eat me, I'm toxic. So the last thing, this is just more me as a biologist, this transition of changing their mating communication is a really interesting model to understand mating signaling, where we have all fireflies kind of had larval light and then gained the ability to produce adult light. And then some branches, at least 5 different points of North American fireflies, they've lost the ability to produce light as adults. This is a great area for us to study. How many of you have heard of like femme fatale fireflies at all? Yeah? My kids know it just because they watch Wild Kratts a lot.
That's it, mine too.
Yeah. So why would you lose the ability to produce light? It seems to be really successful for a lot of firefly species. The thought is that most fireflies have a really potent toxin. If you ate 5 winter fireflies, you would die because they have this really powerful cardiac glycoside that's called elucibuphagin. It's similar to a toad toxin, bufatoxins, and it's luci for firefly, for light, lucibuphagens. And they have a lot of it. More, like winter fireflies have more than most species. So then there's all these other fireflies that don't produce this toxin, but they want the benefits of producing a toxin to protect their babies. So they will sit there in a field like this. They will make a flash pattern that mimics the flash pattern of another species. That male comes in thinking he's about to get lucky, and he gets eaten up. And all that toxin that he had gets sequestered in her body and passed on to her eggs, and now her eggs are protected. So the thought is that kind of immense pressure drove the winter fireflies out of the night and into the day, and they're using pheromones again instead. But we don't have a time machine, we can't figure this out, but we can start to understand, how it works. So that was our kind of question, like how are the winter fireflies communicating? You look in the literature and it was kind of like, well, sex pheromones probably, but there was zero evidence of what that is. So we took on a very ambitious project. Me, a couple of my former colleagues when I was at Bucknell, Sarah and Doug, and then I paired with chemical ecology, synthetic chemist guru Jocelyn Miller here at UC Riverside. And we got some nice funding from the NSF to explore this and how this evolved and kind of the signaling, changing mating communication patterns in fireflies. So our recent paper just came out in the Journal of Chemical Ecology on discovering the first sex pheromone, or first pheromone ever in a firefly species. So I invite you to check that out. But the story is, starts with like, how do you even identify a pheromone? And this looks kind of janky, because it is, but it's also nice and affordable for us. So we can take little mason jars with an aquarium pump in the back here and all this tubing, and we can pass a bunch of air into the mason jar. And right inside the mason jar, we have our little firefly. We have a little piece of screen so she can walk around. This is a female firefly that we're hoping is releasing some sort of pheromone. She's happy enough in that mason jar to start releasing that. So we scrub, this is a volatile olfactory compound scrubber, so just a bunch of charcoal in this tubing that filters everything. Fireflies in here, hopefully releasing a chemical, and then we collect it on a small bed of charcoal inside a tube. And then we use a solvent to kind of pull all that out in a liquid extract, and hopefully we have some pheromone in there. When we have that, we throw it in an instrument known as a gas chromatograph. And this is an instrument that will actually help us sort out different molecules in a big mixture, and they'll come out individually. How many of you have ever done like the ground up like spinach leaves, and you put like the paper towel in, and then all the colors separate? You can get like the yellows and the browns and greens. You can do that with these molecules. So each peak here, just like I showed with the cuticular hydrocarbon barcode, this is much more simpler. We have each peak corresponds to some molecule that was in that jar. It could have just been the background noise of the jar, or something that the firefly was releasing, or maybe the firefly was, excreting something in there. We'll get odors from that too. But then we look at this and we're like, all right, which peak, which compound is the one that the fireflies like? What's the potential sex pheromone? And that's hard to do, right? Is it a big one? Is it a small one? It's not always the same. So what we can do is, as molecules are coming off and being separated here, we can link up a male firefly antenna to the outside. Most of the times when you think about a gas chromatograph, it's linked to a mass spectrometer on the back end that tells you, all right, this peak is this peak is this. Instead, we're taking that out and we're letting the firefly, with its appendage that's been evolved to detect this molecule for millions of years, to be able to pick it up for us. So as molecules come off, we see these peaks didn't really do anything. This is kind of just the noise in the system. And then we see this nice big response from the electroantennogram, which is just a cool word to describe sticking electrodes and picking up the electrical activity on either end of the antenna. And then that points out, like, all right, it's probably this one. We go into that peak, we find this structure, figure it out. It's a lot of work that I'm not going to get into. It was done by a master's student at UC Riverside in Jocelyn Miller's lab. Figure out, is this OH going down? Is it going to the side? Where are these methyl groups branching out? It's a lot of work to get this, even though we knew, all right, this peak is it. So they got this for us, they made it for us, and now we have this big question of, can this work in the field? Right? Is this something that we can, attract winter fireflies with? So another janky trap here. This is a cardboard cake round and a little loose leaf tea infuser ball that we put our pheromone baited lure, which is kind of suspended in one of these little rubber septa, inside, and we have it all covered in a sticky trap to see what can be attracted there. Here's one of my students, Toby, who was at the Bread Loaf View farm, where we have all these, you can see all the sap collection tubing in the background. We're painting that sticky trap onto this cardboard. And then we wait 24 hours later. And then the one that we treated with just solvent has nothing on it. But the one that has this candidate pheromone, it works. We have all these, just after 24 hours, all of these winter fireflies were stuck on there. And they're all males. So now we have a female-produced sex pheromone that attracts just the males. In the words of Khalees, this is the milkshake that brings all the boys to the yard. My wife said nobody would get that anymore, so thank you for that. Yes?
Did you estimate how many of these males were in that area?
That's what we're actually doing next. I mean, some of this is just, we knew that this area had some because I went to this farm before. They're very abundant in Vermont, though. And I think they thrive because they have such sugar sources with the sap. But one of the things we're doing next is kind of looking at the phenology of this pheromone response, because they don't respond to this all the time. If you find a winter firefly now and you wave some pheromone in front of it, it's not in that mode yet. It's trying to find a place to overwinter. So what we're going to be doing this next spring, both here and Pennsylvania, is have our trap locations, look for environmental populations, and then also check the trap and do that every week. It's a lot of work. Yeah. But we want to see, all right, when does that response start? And then we can get into like the gene expression levels. We can have samples that we collect from, they didn't respond to pheromone, and now they start to respond. Is there some sort of receptor gene that shot up during that time? Smoking gun for us. Yeah. So all these males, it's, we're not really big, I'm not a big statistician person, And this is nice and easy because we didn't have anything on the solvent-treated ones, right? So there's very easy stats. Okay. Then the final part of this project we were looking at is like what structures are actually detecting this. And we went back, this is my student Daphne, who's using our scanning electron microscope to start looking at the antennae of the winter firefly. And we see these beautiful hairs, again, very high surface area region. And there's some long ones, and then we were very interested in these ones. They look like little suction cup darts kind of stuck to the side. Here's one kind of blown up. And again, we see all those little dimples. And that's where all the odor kind of moves in and is able to kind of get access to those neurons. So we were interested there. We used a lot of double stick tape to tape down a firefly. Their reflexive bleeding here, that's how they release that toxin if they're predated upon. And we do that. We have a little window here so we can stick an electrode in the eye. Here's an old electrode tip that's stuck in there. And we also have some antennal segments exposed that we can access them with our electrodes. Here's one little nub here that we can look at. Here's the electrode coming in, and we do a very similar thing. Listen to these neurons, puff on our pheromone, and we get this nice response. So with this, we can do different dose responses here. We can see that they're set, kind of start picking it up when we start to get to 1 microgram. And that's when we know now that, all right, this specific hair on the antenna is what's responsible for detecting this pheromone. And maybe we can start looking for these types of hairs across some of these other firefly species to see if we can see similar structures. So with that, we can kind of say, all right, yeah, they do actually use sex pheromones, and we found what it is. It works in the field, and we can move forward there. Now, because like I said before, we are very interested in getting to what the genetic component of this is. So we have the pheromone. Now we want to get what the receptor is. And we're going to use a very similar strategy where I'll take some firefly ORs, put them into our fly system, and puff them. And then individually we can say, all right, we added this firefly receptor. We got this pheromone response. Now we have the pairing here. Excuse me. So the other thing that I kind of teased is this loss of light has happened at least 5 times in North American species, all denoted by these red bars on this phylogeny. And we can start to use this for like biological replicates. Did all of these branches lose light and pick up a pheromone system in the same way, or did they all have to start from scratch and do it differently? And that's where we can start to kind of explore this question and really get some evolutionary data that we can believe in with all these replicates. So that's what we're continuing to do. I like this space, this interaction between the chemical world and the sensory world of the organism. And there's all sorts of different additional questions you can ask in this space. If you ever thought, who cares about insect smell? This is the sandbox that you can play in and where we like to play in as well. So with that, I'd like to thank you for being attentive during this talk, asking great questions. And I want to hear some more questions. These are a lot of the students. This is like 2 Middlebury students, 2 Bucknell students that came up to Middlebury. This is the campus farm. And we were out getting ready for a nice nighttime collection for some lighted fireflies as well. So with that, I'll also thank collaborators at Bucknell and UC Riverside and the National Science Foundation. And I'll be happy to take some questions. Excuse me, we had our first years just arrived, so my voice is waning because I was just yelling at them in the hockey arena all day yesterday about what the biology major's about. So yeah, any questions? Yeah.
How applicable is this work to human pheromones?
So that's a great question. Human pheromones have not been identified yet. People have been looking for decades because you can imagine if you have a human pheromone, all of a sudden, like Old Spice, body spray companies like Axe are calling you up to try to figure out how you could make a product on that. People have done some studies of like like t-shirts worn by spouses and strangers, and you start to try to get familiar odors, but like a pheromone should be something across the whole species. And there has not been anything identified.
Mammal pheromone.
So mammals definitely have pheromones. Like a musk deer produces certain pheromones. You can have a pheromone, they know what it is for like domesticated pigs, and you can like spray it on, and all of a sudden mating happens immediately. But for humans, it seems to have kind of evolved out because it's not as necessary anymore, if it even existed before, right? So it's that kind of thing in humans, or was it just in primates? But in terms of like the relation, one of the things that's really interesting is that our olfactory system, our receptors, are very different from insects. They have the ion channels that are very fast, but they don't have the signal amplification. In cell biology, and you might learn about like G protein coupled receptors, which they activate, and then there's like this whole signaling cascade that amplifies the signal. That's what our olfactory system works with. So we are kind of have a slower response, but it amplifies. How do insects overcome that? They just have a ton of these genes expressed. So the membrane is essentially packed with these ion channels. And the thought is that this increase in speed helps them when they're flying, right? Making that flying decision, you want to be able to turn pretty rapidly as opposed to somebody who's not flying, just kind of terrestrial. Yeah. Yeah, people used to use Drosophila work, and oh, this is a model for humans, and then we discover that they didn't work the same way, and they're like, ooh, Drosophila are a model for mosquitoes. And that's how they're getting a lot of funding now. Yeah?
Can't remember the term, all right, but so when you were talking about the ants and the true queen, and then
The Gamergate.
Gamergate, and you said that these colonies can sort of maintain for
Indefinitely.
Yeah. But so, at what point does that Gamergate become a true queen, or is there like?
She will never.
Okay.
True queens, like while they're actually going through this, like, all right, the true queen, the original queen, the founding queen has died, they'll maintain the worker pseudo queen cycle for the longest time for that nuclear colony. But they will still produce unmated queens and males that will leave. And that's when they will found new colonies that way. Yeah, it's this really, on the scale of like, like a solitary wasp. Like all ants are social. So like you have to go out to wasp, like a cicada killer wasp. They are solitary. Then you have like leafcutter ants that are right there. The Indian jumping ant kind of sits right in the early part of developing eusociality. So we like it as like a primitively eusocial system that is like just stripped down to the bare bones, as opposed to something that has like millions of workers, at least five different castes within that colony. This is a nice simple system that we can start to understand the fundamentals first.
And where are those ants?
They are native to West India, in the rainy, the Ghats region of India. But because of the social structure, nobody's gone to India to get them. All the labs in the U.S. that are working on them just have Gamergate-led colonies, and they just work so well in the lab. Yeah. Yeah.
Do you have contracts with like large farm organizations, with USDA, to like capture pests and analyze other, monocrop pests?
Yeah, so that's like the big extension of this work, right? You can either take this kind of understanding of insect sense of smell to either like ag applications, kind of pest insects and agricultural, issues, or you can take it to like human health vectors, like mosquitoes, certain other disease vector insects, like a kissing bug or something like that. And those people are out there, and especially like the mosquito labs, they have like armies of postdoc researchers that can do this. I like to kind of focus on the smaller questions that we can do in an environment where I can also have just undergraduates doing the research with me. So there we are kind of looking at more fundamental basic questions, kind of like a basic research angle. But I mean, to have these ants, we have to have USDA approval of our ant facility, because we don't want them to escape. So that's our major connections. But that's a way that a lot of people will go they'll take some sort of pest, like maybe something like the emerald ash borer. Yeah, you can use that. Same thing with like what I showed with like the apple orchard. Like once you understand the pheromone, you have this unique angle to just that species, and you can control it pretty effectively. Yeah. So if we had something for like the Asian longhorn beetle, which is not here, but it's flirting with being in this area, invasive pest, we can use that to catch a bunch of these and trap. That might be a great way to reduce and maybe eradicate it in the area.
So this is actually to treat any type of insect?
If they use a pheromone, something like a cicada, they're not using a pheromone, they're using an auditory signal to talk to each other. We hear those a lot right now.
What about locusts?
Locusts, it depends. There's certain ones, like the ones that really kind of come in and decimate, they get like big swarms. There's really good research there where like you can actually have them in the lab, and it's like once they get dense enough, that's when they become the gregarious phase, and that's when they start to swarm and fly. And you can actually, as a researcher, like just poke their back legs a lot, and it's the contact between all the other locusts around them that causes them to transition to that phase. So that's more of like a tactile thing. But if there's something that's like an aggregation pheromone, that's what the bark beetle people in Sweden were using. They tap into the aggregation pheromone that brings both males and females together. Then you can have a really powerful control strategy. Yeah.
So when you showed those videos of, the one ant sort of trying to keep the other ant from doing bad behavior, non-acceptable behavior, so do they gang up, or is it really just like one that sort of takes the lead and the others are sort of just like hanging back?
It kind of depends. For situations like that, usually if they're like one is doing the policing, but sometimes you can get like really good pictures of like, you can drop an ant from another colony in there, and all of a sudden there's like 4 or 5 ants all grabbing the offending individual from different sides and like, really, doing aggressive policing behavior. It's very similar to like paper wasps, which they don't just kind of have dominion over the colony. They have to do a lot of aggressive behaviors towards each other. But when you have like the antennal dueling back and forth, that kind of, both of those individuals kind of get pumped up and they kind of elevate their status. But when that happens, you have other ants that are not engaging with that, and they'll kind of lunge and bite and grab the other ones, and they kind of like take them out of the equation. So there's like a whole, people have looked into like the game theory behind who becomes the new pseudo queen in that process, in those behavioral interactions, and try to like model it with like, all right, dueling is like a +10, and then getting bit is like a -25 or something like that. Yeah.
Do you think it's a large field? Do you have a lot of collaborators? Seems like you have a lot of students.
Yes. The nice thing with where I've kind of positioned myself, there's a major field is like looking at insect genomics and trying to look at these things. So like people will be like, oh, here's all the olfactory receptors, and we've identified them and annotated them, and then it kind of stops. I've situated myself and like the work we do as like, let's start to decode how those receptors function. So that leads people to reach out to me. Not many people do it. And like, all right, like I have somebody in Arkansas who is looking at a butterfly mating system in which the male produces the sex pheromone for the female. And they are sending us data and sending us receptors to do that. I got started on Fireflies because Sarah saw me give a seminar at Cornell when she was there and put herself on the schedule to talk to me about Fireflies. A dream of hers that she's had, and like, we need to get to this, and now we get to do it, which is kind of fun. So it's a big enough field. There's more focus here on mosquitoes and other human health pests, and also just Drosophila is like, all right, let's understand the model or insects species. In Europe, there's a lot more focus on this as on insects. Like there's a big international symposium on olfaction and taste, and like Europe brings a lot of the talks I like to go to because it's focused on insects, for insect's sake.
What about African nations, where this could be like so valuable?
Yeah. So Bill and Melinda Gates, when they were still together, had the Bill and Melinda Gates Foundation. They funded my like graduate mosquito work, because the idea was that they might be able to put some money into a product that could be given out for free or very affordable in Africa, and then leverage those costs by having some sort of commercial product in the U.S. and other wealthier areas. But there's a lot of work being done historically and problematically. It was kind of being done in African countries, and they just take the research out and exploit that population. Now there's more work with these communities to get these situations managed and sustainably, where like tsetse fly is a big disease vector, and they've eradicated that in certain islands off the eastern coast of Africa, because they found out that tsetse flies really were attracted to like the smell of cow urine, and for some reason they liked the color blue more than other things. Who knows why? But they would have like these big oil drums and put them horizontally, and then in front of that was an electric net. The tsetse fly comes in very fast, hits the net, and dies, and then you have just piles of it. And they eradicated tsetse flies, which is just a powerful thing to do. Yeah, with ecological considerations as well. But if you eradicate the species to take out, the disease that they transmit, then you can reintroduce them, and maybe the disease goes away.
That is it for time. If you have more questions, please feel free to come on down. Let's thank our